Our next panel is called It Takes a Village, Scientists, Communities, and the Co-Development of Eco-Technologies. Engagement is often promoted as essential, especially for decisions that have community-wide impacts, but that people can't necessarily opt out of once they're made. And so this panel is going to explore case studies of scientists engaging with communities to inform the use of eco-technologies, including gene drives, but not limited to them, that could control and even eliminate entire populations of disease-carrying mosquitoes and invasive species. We have on the stage both scientists and engagers, as well as representatives of communities from across the world that are engaging on these topics. So we're thrilled to introduce this panel. Our moderator is Megan Maltini of Wired. She is a staff writer where she covers the ways in which leading edge technologies like CRISPR are transforming science, health, and society. Her work has appeared in Popular Science, Discover, Undark, and Ion, and she studied molecular biology herself and ultimate frisbee at Carleton College and has a graduate degree from UC Berkeley. So thank you, Megan. I'll turn it to you.
Thanks, Julie. So as I was sitting in my Lyft going to my hotel last night, my driver asked me why I was in town from San Francisco, and which inevitably led to kind of the 1-minute pitch on what CRISPR is, with all the requisite analogies to scissors and word processing. And when he asked what could this potentially be used for, I kind of chickened out and just talked about crops and medicines, which of course is not the subject of today's panel, but explaining CRISPR's potential for shaping eco-technologies, including gene drives and other methods for adapting and controlling and even eliminating populations of species, was just going to take longer than we had left in the drive time. But it really made me realize firsthand just how Herculean a task it is to really engage on this complicated and controversial topic. So I'm so excited to have this special group of people who've flown in from all over the world to let me pick their brain about it for a whole hour. And so I just wanted to start by letting each of them introduce themselves and the organizations that you represent. So just take a minute or two to kind of tell everyone who you are, and then we'll go from there. Start with you, Mel.
Kia ora koutou, ko Melanie Mark Shevalt tōku ingoa. That's my name is Melanie. First, let me acknowledge the Indigenous people of this land, so kia ora and thank you to them for welcoming us here in these great lands and these great hills. I am a social scientist at Lincoln University, first and foremost, but I'm also the CEO of an organisation called Te Tira Whakamātaki, which is loosely referred to as the Māori Biosecurity Network, though it doesn't mean that. It means the watchful ones or the vigilant ones. And obviously our focus is on protecting our environment, and especially our biosecurity system in Aotearoa, New Zealand. Kia ora.
Kabai Edward is my name. I'm a local council chairperson of Uganda Research Institute, where an insect ray is going to be built, or it is built actually. And we've been engaging with communities to pass on information to the communities and trying to find out why exactly malaria is still hard to eradicate. Thank you.
Hello, my name is Carrie Filer, and I currently work at the Martha's Vineyard Regional High School. I consider myself an ecologist, a parasitologist, a molecular biologist, but right now my role is primarily educating the high school students on Martha's Vineyard. I am on the steering committee for the Mice Against Ticks project coming out of MIT. And I represent a representative for the Chilmark Board of Health. So I've been highly engaged with the community in trying to inform them about the Mice Against Ticks project, so that one day, if there is a vote, they will be informed enough to make a decision.
I'm Kevin Esfeld. I'm an assistant professor and leader of the Sculpting Evolution group at the MIT Media Lab. I am fortunate to be at the Media Lab because even though I am a scientist and biotechnologist, I have the freedom to do things a little bit differently. And because I am a techno-optimist and that I believe that the future of our civilization will primarily be determined by the technologies that we invent and the wisdom with which we deploy them, I am not remotely confident of my own wisdom to choose a decent path forwards. And so what we've been doing is going to communities before we develop the technology and asking them, is this a good idea at all? Is there particular concerns or suggestions you might have in terms of the ways that this technology should be developed in those critical early stage decisions that could help prevent us from making mistakes that could be deeply damaging, whether to the environment, although frankly I'm not so worried about the environment as I am about our culture. Because as was previously discussed, gene therapy was set back more than a decade by a single tragic death in a clinical trial. As the first person who told the world what a CRISPR-based gene drive could do, I feel personally morally responsible for all the consequences of that technology. And to the extent that there is an accidental release, I think it would become extraordinarily difficult for representatives of local communities to make any kind of argument in favor. So I am very concerned with changing the incentives governing scientists to encourage that kind of local community involvement, because we don't have the expertise to help determine whether or not these technologies are a good idea. And I'm not sure how effective it is to talk to the world at large. So we've been working with, locally, the islands for mice against ticks, and it's not really a formal project yet because the Maori have not established any kind of formal engagement with us. But insofar as we are developing gene drive systems that are intended to be local and not spread indefinitely through the entire population, we're interested in learning from them as to how we should go about doing it. And we've already screwed up a number of times, so that's something we should talk about.
We'll get to that later.
My name is Delphine Tizi. I'm from Europe, and I work for a non-for-profit consortium called Target Malaria. And what we do is we try to co-develop with many other stakeholders a gene drive solution for malaria, which, as people might know or not, still kills about 500,000 people per year. And so we're trying to see how we could reduce the number of mosquitoes that carry it. And my role in it is not a scientist, but I work with a great team in countries to engage the communities, engage civil society, religious leaders, governments, policymakers, and to try to see what is their input in the development of this technology. And yeah, and our project's been around for 13 years, so it's a very long-term research and long-term solution. But we believe that, as Kevin just said, that the conversation needs to start early.
Great. Thank you. Well, I know we have people from lots of different backgrounds in the audience, so I was hoping we could just start simply by defining kind of what we mean by eco-technologies, especially because, as you mentioned, you've been doing this longer than gene drives have been around. So maybe Kevin and Delphine, can you guys kick us off by just giving us kind of that umbrella level definition?
I start? Okay. Actually, it's the first time I hear the term eco-technology, so I'm going to give it a try. I mean, in our case, what we're trying to do is to find a novel way of targeting the vector of malaria. And the way it's novel is that, unlike a lot of the existing technology where you can make an individual decision, whether you want to spray your house, whether you want to spray yourself with an insecticide, sleep under a mosquito net, the use of gene drive requires a community decision or country or even more regional decision. So there are gene drive and other area-wide intervention require a different type of engagement and a different type of dialogue and of acceptance. And this is what we've been working on in very short.
So, to be clear, when we edit the genome of an organism, or we simply selectively breed it as is traditional, we almost always reduce its ability to reproduce in the wild. And that means that human-altered organisms simply cannot compete. Natural selection almost always wipes them out. There is no known case of an engineered or selectively bred organism outcompeting its wild counterparts in its ancestral habitat. But gene drive is a completely ubiquitous natural phenomenon. That is, it's present in just about every species we've ever sequenced. Most of our own genome is broken remnants of gene drive systems. And it's defined as a phenomenon in which a genetic element spreads even though it doesn't help the organism that is carrying it. Instead, it distorts inheritance in its favor, so it can spread regardless. And so by harnessing gene drive, which has been a dream of scientists for a very long time, and the mechanism that is most promising that CRISPR uses was actually postulated by Delphine's colleague Austin Burt back in 2003, which was the dawn of target malaria. And CRISPR has just made it possible to do that in potentially many species and not just mosquitoes, with a lot of effort. That means that we can probably do it. We can probably edit wild populations. And I'm concerned that we do that thoughtfully and using the appropriate tool. Because to be very, very clear, for malaria, we need the kind of gene drive that spreads indefinitely, that would spread not around the world, but would spread to all of the major malaria vectors in Africa to have a hope of eradicating malaria. This is at least my understanding from the models. I don't study malaria specifically, but it's hard to imagine, other than a perfect vaccine, a tool that would let us get all the way there. So we need that for that problem. But I'm concerned that scientists who might be very enthused about using gene drive for other problems might make a mistake that could then impair that effort, which in my view is by far the most important application of gene drive. So for other problems, I would prefer that we focus on developing localized solutions where the engineered genes cannot spread indefinitely, but rather rely on a number of mechanisms, either depending on the frequency of the population or they run out of genetic fuel and stop, would allow local communities to make their own decisions. Because I'm going to have to disagree with all of you. I am a huge fan of local community governance because there are so many cases where they know things that we don't.
Well, that's a great lead-in. So I want to hear from Mel and Edward and Carrie. Can you guys give us a sense for the scope of the problems that are happening in your communities and which technologies in particular you're most hopeful about in terms of harnessing them to solve some of those problems?
I'm happy, to start off. So on Martha's Vineyard, Lyme disease and other tick-borne illnesses are such a huge problem, and it really is quite terrifying, I think, that I can't even walk from my front door to my car without being fearful of getting bitten by a tick and coming down with some tick-borne disease. We have anybody with young children on the island are doing tick checks on a nightly basis. The ticks that I find are not the ones that scare me, they're the ones that I'm not finding. And the symptoms can be debilitating, they can last a lifetime, and sometimes there are no symptoms right away. And so the bacterium can kind of hide out in your body and wreak havoc. So it is a really big deal on Martha's Vineyard. There has been talk for a long time about what we might do as a solution. We've talked about, culling deer. Some people spray their yards, chemicals, this, that. But it wasn't until a couple years ago when Kevin and Joanna reached out to me about this project that I got excited for the first time that there might actually be a solution to this problem, which is why I immediately jumped in as quickly as I could to try to help out.
There are so many outcries out there about malaria being the biggest killer disease, especially in Uganda. But little is known about the administration, the extent to which people monitor the medicine. In the villages or even in the urban areas, people behave differently. When somebody takes a dose of malaria, for instance, he may or may not complete it. And once he feels like he's fair, he abandons the medicine. This crops up many issues, like drug resistance, for instance. And you find somebody suffering from malaria again and again. But the lab people or the scientists have not gone further to ask why this is happening. There's little information that is passed across to the victims. And I think if there was a technology which could administer or reduce numbers of infecting mosquitoes, I think people would be happy. Because in most cases, you find that if, for instance, you force a kid to take the drug to the end, you might be nagging, and they will not like it. So instead, gene editing, the way I conceived it, I think it will be a very big solution to this.
Kia ora. So I guess for Māori communities in Aotearoa, New Zealand, one of our biggest issues is the loss of our sacred species, so our taonga species, our trees and our birds. Especially New Zealand never had predators originally, so we were an ecosystem built on large trees and birds, predominantly birds that don't even fly. And of course, colonisation and globalisation has brought in numerous pests, and we have a huge issue with biodiversity decline. And as the indigenous people of that land, our whakapapa, which I think the best way to explain that is genealogy, so it's the English interpretation of it, but it's actually about our taxonomic framework, the way humans and non-humans are related to each other and the interactions between them are affected by biodiversity loss. So by losing birds and trees, we lose part of our culture, we lose our language, but we also lose the very essence of who we are as tangata whenua, or people of the land. So we're looking for technologies, obviously, that can help with predator eradication. But obviously those technologies, as Kevin has alluded to, have to be mindful of the fact that those predators that we want to eradicate, for example, possums or rats, are also sacred animals to other Indigenous people, especially the possum, which is sacred to Aborigines in Australia. And so we're very mindful of technology development that helps our environment and helps our people, but has consequences for other Indigenous people around the world.
Great, thank you. So I want to get at this question of, you know, as Julie pointed out, that this is technologies that we're talking about that individuals can't opt out of. And so I want to get at this question of who gets to decide who has a right to have a voice in that debate. And I want to hear from all of you, but I was hoping maybe we could start with Kevin and Delphine to talk about some of the processes that you've set up to engage with local communities, and then we'll go to Edward, Mel, and Carrie to hear about how well those processes have or haven't worked.
Go first again.
I go first. So we've started engaging about 5 years ago when we started a collaboration with the 3 partners in Africa, in Mali, Burkina Faso, and Uganda. And it was a big learning curve about who we should engage, how we should decide. And at that time, there were not that many precedents. So we looked a bit around other technologies that are not gene drive based, but that have been releasing mosquitoes or working on similar approach. And we looked at Wolbachia-based technologies for dengue and GM insects from Oxitec. And from this, we learned that you need to be inclusive, but you also need to see what is the right model for where you're working. So we looked a bit at what they had done in their context, which were mainly Asia and Latin America, which were quite different. And so what we decided to do is, with the partners in country, was to see what would be relevant to African societies in terms of decision-making mechanism. So to give an example of a different country, so Edward can speak about Uganda, but in Burkina Faso, our team spent about 8 months to a year to talk to the communities about who should we involve, how they wanted to make a decision, who would make it, should we go for votes, and what would be the threshold, which is always the big question, or should we mimic the traditional mechanism to make decision? So they had this long discussion. We did bring some of the values of the project in terms of inclusiveness, and we wanted to make sure, for instance, women who are not always very represented in these communities would be part of it. And so in a lot of the place where we work, the communities have built representative groups that represent the different parts of the community, which are kind of our first interlocutors with who we discuss about the next steps. And they are also the one representing when we want to make a decision. So they're the one who say, as a community, we've discussed with our traditional way of discussing, which we're not always involved in, and this is our decision. And in the project, we have made, I think, a similar commitment to Kevin's, was that if local communities where we plan to do the next step of our work were not agreeing with this work, we wouldn't implement, we wouldn't go ahead against their will, even if we had regulatory approval. So we believe that the the acceptance from the community is as important as the regulatory acceptance, which obviously is a legal requirement. So I think it depends of the context, and I guess we have representative of very different contexts, but this is what we found was relevant for Africa. And then we also engage at the global level, which is what I'm more in charge of. And there, the work we're doing is to make sure that the global public, which I guess is what we have today in front of us, is part of this discussion, but also acknowledges that malaria being mainly a problem in sub-Saharan Africa, the sub-Saharan African countries and communities are the one to lead this discussion and the one to make the decision.
Edward, can we go right to you right away? Can you talk about your role kind of as a go-between between Target Malaria and the local communities in Uganda?
Oh yes, I was engaged by Target Malaria mainly to mobilize and help them get onto the communities deep. We organized meetings where the staff of Target Malaria talked to them. There are some barriers like the language and other things, scientific terms to be passed on to the local communities. They are not very scientific, but we got them in groups. We had communities like the churches, you get women, you get youth, you get the men separate, and they can talk at ease. They can raise their concern, and the staff of malaria, Target Malaria, can easily answer. Then we were introduced, we were requested to know about the construction of the insect tray. Of course, this was news. They are going to construct an insect tray, but of course you can't click at first time. You have to think about it. There are so many fears allayed, and you have to explain a lot of things. Imagine a case where the beneficiary wants to be paid before he gets the knowledge. I mean, it is monetized, something like that. Somebody to come and attend a meeting, you need to induce him or her. It is very difficult. So you have to have a way of reaching them and passing on to the information.
Mm-hmm. Can you guys talk about how local knowledge and culture has shaped some of those processes and how some of those voices have changed the work?
Yeah, so for us, a very clear example of how what communities say shapes the project is when we discuss about this technology, and I think the previous speaker before mentioned that, there's always this question of ecosystem and what would happen if you reduce. So we're not planning to eliminate all mosquitoes. It's one species out of 3,500 species in the world. But still, what happens when this one species is reduced to a lower number? And this is the one question we get. And you get it in villages, but if I go and engage ambassadors at the UN, I get the exact same question. So we thought, okay, this is the number one question we get. And so how are we going to respond? So if I ask my scientific colleagues, they're like, Anopheles gambia, which is the species we work on, is not a keystone species. This is known in ecology. There are precedents of it being reduced thanks to bed nets and other tools on specific areas. So we know there was no ecological disaster in these areas. But yet, it hasn't been specifically researched. So we went to see some of our funders, and we say, this is the number one question. It's an important question in terms of public trust in this and being responsive to public concerns. So we just got a grant to start a big study using DNA barcoding to understand what is the ecology of Anopheles gambia and to respond to these very precise concerns from communities. So this is the one example of how the community concerns shape the work we do, and in very practical terms.
Do you have anything else to add to that, Robert?
Not now.
All right, well then let's move ahead and talk about Lyme disease.
So for mice against ticks, this was just well, let's be frank. I was concerned because of CRISPR gene drive. And I was interested in the question of, is there some sort of smaller scale intervention in which we could work with ideally a local community, so we could go there on a regular basis, and that on a project that might make a difference to people. And so tick-borne disease, of course, in the Northeast is the thing. I mean, we always say that in the West you have earthquakes, and in the South you have hurricanes, and in the Northeast we don't really get either of those to any significant extent in New England. No, our natural disaster is Lyme disease. So this idea of encoding the antibodies is something that is sort of clearly doable on an engineering basis. Like, there's nothing really scientifically all that novel technologically about doing this. It's putting together pieces, techniques that are known to work. So we were reasonably confident we could do it, at least for the anti-Lyme. Anti-tick is a little bit less clear. But how do you go about it? So we had a direction-setting meeting in which we invited local island stakeholders, although I really hate that word, and other ecologists, and just to find out, and medical doctors and science communicators to say, is this going to work? Could it work? And how should we ask the communities to guide it? And they said, broadly speaking, go talk to the boards of health at an open town meeting, advertise it, people will show up, they will yell at you, you will find out. And that's more or less what we did. And overall, the response was very positive, but we were very careful to put a number of options in front of them. Number one is, do you just release a lot of mice on these islands, which of course would work for islands because they're islands. If you release enough copies of mice that have a dominant Lyme resistance gene, then on average, future mice will all inherit a copy, or at least enough of them that you're going to knock down the disease by a lot. Or you could release a lot fewer mice and use some kind of drive mechanism. But I was very clear that building a drive that would not spread to the mainland was something that was very much in development and uncertain. And we would have to encode CRISPR in the mice. Now, this is something that the communities, by and large, did not want. That is, they strongly preferred that we only use white-footed mouse DNA in engineering these mice. They didn't want us to bring in any foreign DNA. Now, to a scientist, especially in biotech these days, that's sort of like being asked to work with both hands tied behind your back. But it's doable, and it's what the community wants. So that's exactly what we're doing. That means no drive. That does mean releasing a lot more mice, which has now become the number 2 concern. The number 1 being, are there going to be ecological effects, exactly as before? And the answer to that is, we have no real confidence in our ability to predict ecological effects. Because yes, there have been relevant studies, but this is an ecosystem. Biology is complicated. It's complicated enough in individual organisms. Trying to figure out genetic screens for psychiatric disease, that's insanely complicated. In many levels, it's worse on an ecosystem level, because at least individual organisms have been shaped by natural selection. We expect there's certain functional constraints. Ecosystems, not so much. So the only real answer is you try it on a small scale, see what happens, and then use that information to decide whether or not to move forwards. So thus the small uninhabited island field trial was a key factor that was identified early as would be necessary. And then this question of immunize against Lyme or against ticks, or both, or don't do anything. And so we raised these at the town meetings, and they said, okay, 100% mouse, but if you're going to do anything, do everything. And we would love to have fewer ticks. So that's what we started doing. This was a project where the options available were very well defined from the outset in terms of these are the portals that we could walk through, and which one does the community want? And that, of course, can change later on, but that's why we set up the community governance, although that was largely determined by the communities themselves. And sorry, Carrie, I'm going to have to admit that was largely a Nantucket thing, primarily because we talked to them first. And they just said, here's how you do it. Steering committee, you have an independent data safety monitoring board to look at the field trials and ensure that everything the community asked for is done in terms of safety studies. But we left that entirely up to communities because governance is a civic question. It has to be left to the communities. It's not really our place to say anything at all about how a community decides to govern a project like this, because the shared environment is a civic matter.
And Carrie, can you talk about how well those processes have worked, kind of from your point of view?
Yeah, so I have been fortunate enough to come to a couple of the town meetings, the Board of Health meetings, where Kevin has presented the project, and I think overall the community is very excited about this project and the fact that something could be done about the problem that we have on the island when it comes to tick-borne disease. I have a really unique opportunity to engage with the community as a high school biology teacher. And so I have been, my students actually, we have been studying the proposal, and we've been kind of delving into the science behind the project. And the students are excited, and their parents are excited. And I think the community in general will listen to the students as they talk about our involvement with the project. We're able to go to the MIT Media Lab and see some of the labs where the research is being done, and that was so meaningful for us. And later on in the month, my students will actually be giving a talk at one of our local libraries, discussing the project with their friends and their parents, and probably a whole different group of people representing our community, completely different group than what Kevin would get at one of his meetings. So we're able to reach a much more diverse group of people. And the other work that we've done this semester is creating an informational pamphlet for our community, because so many people want to know more about the project. They just don't quite know where to go and who to ask the questions to. So we'll be debuting this informational pamphlet to our community at this talk that we're giving on June 19th. But I would say the community is excited about it. I talk to people who I think will have a huge problem with this type of project, and because they're either, I think they might be an anti-vaxxer, or they might be, I know they're anti-GMO, but then when I explain the project, they're like, oh, well, that's great, that's awesome, 100% mouse, so it's not, it's not exactly, it doesn't alarm them when I actually explain the project to them in a way that a very general way that they can understand. People get incredibly excited about the potential. And so I'm excited about seeing it hopefully move forward.
Do you have anything else to add, Kevin?
Yeah.
Well, for my part, I'm excited by the prospect of involving the local skeptics. Because on both steering committees, actually, we have sort of a appointed vocal skeptic who, and it was sort of luck that they were nominated, but it's the skeptic's job to channel the voices of people who are uneasy with the project. Because it was very clear, in particular from the first Vineyard meeting, where one of the local Lyme support groups just turned out in force. And yes, we had 120 people in that hall, and probably 80 of them had either had Lyme disease themselves or one of their family members had. And so I could readily imagine it would be uncomfortable to stand up in that environment and say that you're deeply uneasy about this idea of engineering the mice, just because there is so much enthusiasm in the room. And that worries me, because we know that local communities can come up with technical concerns that we haven't thought of, because it has happened. So the best and easiest example is from a town hall where someone said, wait a minute, if you immunize the mice against the ticks so that the ticks don't like the taste and fall off, then that first year when you release all those immunized mice, doesn't that mean there's going to be more infected ticks that could then bite a mouse, fall off, and then bite a person? And I turned to Sam Telford, who's our lead ecologist, and we looked at each other, and I said, we didn't think of that. That's why we're here. And the answer is, adding the immune mice doesn't have that effect. But anytime you reduce the native mouse population, the native hosts, you are going to have that effect. And so one of the original ideas was maybe the island would want to remove as many of the native mice as possible, drive it lower than the natural low point in early spring, and then release fewer engineered mice, but maybe they're not going to want to do that because of this question that was raised by a community member who did not have a terminal degree. So that's why we're here, and that's why we have the vocal skeptics, to ensure that any community concerns that people aren't comfortable raising in public or by approaching us directly, they can approach their local skeptic on the steering committee, and know that their concerns will be raised, because we want to hear all of them.
Great. Well, so after we've talked about some kind of successful stories and hearing, having this bi-directional dialogue, between the research community and the local communities, I want to make sure that we get to Mel and talk about the question of how researchers interested in ecotechnology should be thinking about building respectful relationships with Indigenous communities. And since the two of you have had a somewhat public exchange on this issue, I'm going to put you both on the spot to discuss some of those missteps as we talk about how Daisy Drive development might go forward in a Maori co-governance structure. So I wasn't going to read any quotes from that, but maybe you guys could fill in the room on kind of how that went down and what you've learned kind of since then.
Do you want me to give the?
If you want, you can start.
All right, so basically I screwed up when trying to fix an earlier screw up on my part. And that earlier mistake was in our original description of CRISPR-based gene drive, in which I made a very pretty figure suggesting all the potential applications. And of course, the figure is a simplified version that doesn't have any of the caveats, which are all in the main text. And so to the extent that some individuals in the conservation community in particular are very enthused by this idea of gene drive to control invasive species, they didn't necessarily gather that all of the caveats, namely that the self-propagating version would almost certainly be moved to other regions, because most things that are a pest somewhere are also an economic pest, certainly rodents. So it would almost certainly spread to other areas, and that means that if we had a one world government, we could totally take care of it, technically. But in our world, that's probably not going to happen, and that could have a lot of deleterious effects. So we did some additional research and modeling showing how invasive the spreads forever version is, and essentially said we need to be careful and think carefully about which applications, like malaria, absolutely need that, and which ones we should instead take a step back and think about new solutions. And so I wrote with Neil Gemmell, who's a colleague in New Zealand, a piece for PLOS Biology on how we needed to be cautious involving development of gene drive, particularly for conservation, and ensure that we only work on the localized versions. And the problem was that my first visit to Aotearoa was after we submitted the paper, but before the revision stage. And so I first was introduced to Mel and her colleagues in between, and I thoughtlessly did not consider the political implications of publishing an article on gene drive that, because Neil was local to New Zealand, we used New Zealand as a case study. But of course, there were people who had taken sides on gene drive self-propagating that meant that that article, which had a lot of media coverage, had local political implications. And that jeopardized our partnership, because one of my goals is to ensure that the Mātauranga Maori, the traditional knowledge of the local ecosystem, which is not reducible to Western scientific terms, probably can't even be translated into those terms, I want to be sure that that is used as a parallel safety study. That is, determining whether or not this is a good idea. And it's very much the sort of thing where we're going to rely on you to tell us, is this a good idea based on what you know? And we're just going to take it completely on faith, because you probably can't explain it in terms that can be reduced to Western scientific practice in any meaningful way. So if you say it's a bad idea, then it's a bad idea, and that's that. And I think that, broadly speaking, when we're developing these technologies to alter the shared environment, we really need to do that wherever you do have this local, especially First Peoples knowledge base. And so ensuring that that model is adopted in Aotearoa was a primary goal for, I think, both of us. But I jeopardized it.
Mel, can you tell us a little bit more about that from your end?
Yeah, I guess that's a really good summary, but basically when Kevin first came over, one of the very important discussions we had was around co-governance and sharing. So we have a treaty in New Zealand. I know in North America you have lots of treaties, but we have one treaty that covers most of our tribes, and that affords us three things primarily with the government. So we would never say we ceded sovereignty. What we did is we agreed to a partnership, but a partnership that included participation and protection of our taonga and our species and our land and our rights. And so we talked about co-governance and what partnership meant, and said that exactly as Kevin said, any project going forward needed to run parallel systems, our knowledge, mātauranga Māori, with Western science and in a true partnership model. That scares a lot of people in New Zealand still. There's still a lot of agencies, ministries, government organisations that don't want to have the discussion about partnership and what that looks like with Māori. And so for us, it was really positive to have, someone come along and say, we want to talk about co-governance. But that partnership to us, or to me in my mind, meant that we discuss everything like partners do. And like I said, what you're supposed to do with your husband, not happens in my house, but what you're supposed to do with your partner is, discuss and have an agreement. And we hadn't had that. We had an agreement that we'd work together, and then we had an article that came out and did have political consequences for us. So, we also have in the Indigenous space and in a lot of community spaces, we have extraction of our knowledge. So on top of, promises for partnership, we have people coming in and extracting our knowledge continuously and then excluding us from the decisions about how that knowledge is used. And that is in the ecotech space as well. Right now we have a phytophthora killing our sacred trees, our Kodi trees, and there are numerous researchers in there extracting Indigenous knowledge to see how they can potentially save the trees and cure the disease, but none of that goes back to the Indigenous people. So on top of all the issues, we had, a promise of partnership that was then, it wasn't a huge mistake, but at the end of the day, it was a reminder that there are consequences for the communities that are left behind when the decisions, when promises are broken, really.
So how have you been able to move on since then?
Kevin's come back and he's done his apology tour.
Is this part of it? Are we on it right now?
It's ongoing.
It's happening.
And that's in part, it has to be ongoing. So I went back and they generously invited me back through Te Harenga, including Doug and Thea, who are here in the audience, are representatives of Te Harenga there. And Mel's Biosecurity Network also hosted us generously I shouldn't say we. This was me. I thoughtlessly did not send that draft to you when it could have been changed. Yeah. And I just didn't think of it. And it's a great example of why we need to consult local communities, because here I was blundering around doing something that had local implications on local politics, the local political ecosystem, that I was just entirely ignorant of. I didn't know how many people in different branches of the New Zealand government, for example, had a stake in this and the delicate, what the current status of co-governance was. And I put it in jeopardy just because I wasn't thinking about it, because I didn't consult the locals on matters where they know more than we do.
And to be fair, as researchers, and I'm a researcher as well, as researchers we get very excited about what we're doing. And I understand where Kevin came from. We get very excited about what we're doing, and we move ahead. We're trying to progress in a system that's naturally slow anyway in the academic field. But as Kevin said, one of the keys to engaging with communities is, one, having projects that are led by communities and that communities want, but two, is mindful of the whole environment and all the other things that are going on in that space.
And can you give us a sense of what it really means to invest in these communities for the long term as researchers to avoid this kind of drop-in extraction model?
Yeah, I guess one of the biggest failings we have in the New Zealand system is that we don't front load research, and so it takes researchers a long time to build long-term relationships and to invest in that relationship and those conversations. And so quite often research, especially in Aotearoa, is extractive because that's the nature of the system. You can't do anything until you get funded, so when you do get funded, you've got that small window of time where you have to get in there and get as much as you can, to produce your outcomes so you can get your next funding bid. But that's by and large not the way our communities, it's not the way our communities want it. And as our Māori economy, so as our Indigenous economy grows substantially post-settlement, as our treaty claims are settled, and we grow faster than the New Zealand economy, then there's pushbacks, from our Indigenous communities saying, well, we won't tolerate that anymore. So building long-term relationships is important as a researcher because obviously it helps guide your research. It means that you have those, as much as we hate the word stakeholder, you have those stakeholder relationships so that you can get funding applications, but also it means that you're doing work that's meaningful for the communities, that's driven by them. And as Kevin has said before, you never know what you'll find from a community. So quite often, especially in the Māori space or Indigenous spaces, as Melissa would have alluded to before, you quite often find those gems of information or knowledge, or discover things, or you rediscover things that our elders already knew.
Well, I feel like we've had a couple different avenues to get into talking about incentive structures and how those need to change to be able to, move forward with eco-technology development. It seems like hearing, regulators want to see some kinds of safety studies, and scientists have the kinds of studies they want to pursue, and communities have the kind of, research that they feel is, central to feeling safe, having these technologies progress. So, can we talk about those conflicts and kind of how each of you in your roles are striving to kind of align them, and what are the biggest structural barriers when we talk about incentives? And that's kind of an open-ended question for the group.
I'll start because we face that a lot because, as I say, we're now quite an old project. We've been here for 13 years. And you can see the differences in terms of all what we need. So our consortium is now about 140 people, and most of these people are academics, researchers. So the way that academic career is constructed, they need to publish. So they need to publish new things. And on the other side, we have questions from regulators, from communities. That might not be really exciting science. So to give an example, we do get from regulators or from communities the question about could the species we're targeting, Anopheles gambia, mate with a completely different species, let's say Culex, completely unrelated. When we ask the colleagues, the scientific colleagues, like, yeah, no way. It's like asking me if a monkey could mate with an elephant. So there's no point of writing a paper about it. But for the communities, there is a point because it is a question they have. And there you have the issue of if you want to publish something like this, or if you want to make it available, what's the incentive for the scientists to do it? So I'm currently working with a very large group of engagement practitioners about how do we do engagement for this kind of technology, what are the kind of guiding principles we should follow, and so on. And we're struggling to find where we would publish this because it's not exciting science. We don't want to publish it in social science journal because our audience are scientists, technology developer, but the big scientific papers, our paper doesn't quite match the format. So it's all of these challenges that as you're doing really exciting science, you're also building all these other new paradigm in terms of, regulatory needs and engagement needs. So it questions a lot the existing framework.
I admit this is my pet peeve, because why do we do science the way we currently do? That is, if you were designing science from scratch to serve the people, would you really incentivize everyone to keep everything they are doing a secret from the wider world and even their own colleagues and competitors? I mean, sure, that's a competitive spur, but that's not a very efficient way of going about it. No one designed the scientific enterprise to be so bloody secretive. It just evolved that way. That's just the way the institutions evolved, because until comparatively recently, you couldn't efficiently share your research plans with everybody else. Now it costs literally nothing. But the incentives all point towards keeping your cards close to your chest. Because if you share your bright idea and one of your competitors gets wind of it and does it first and publishes, they get all the credit and you get none. And even if you're tenured, you're still responsible for your students. And if they spend 3 to 4 years working on something and then get scooped, that could be it for their career. So the incentives are broken, overwhelmingly broken in my view. And you really have to take a step back and think, why are we doing it this way? Is there any reason we do it this way? And there really isn't, but at the same time, you can't just unilaterally change the whole system. That would be unwise. We kind of depend on technology. I mean, I'm nervous about the ways that it could go wrong, yes, but we absolutely need it, and it is the reason why health and well-being have improved so dramatically over the last century all around the world. And we can't very well stop either. We are not sustainable from the conservation perspective. We are using too many resources, and if we want to continue even as we are, we need new advances in order to make that happen. So we can't stop, and it would be reckless to change a system as complex as science all at once which is why I would love to see us run a field trial, multiple field trials of new incentive structures governing how scientists behave in small, suitable subfields. So to me, the most exciting application of Gene Drive is the potential to use it to change, to at least trial a new way of how science should be done. That is, pre-register your experiments before you do them to give everyone a voice in those early stage research decisions that are intended to ultimately affect them with no possibility of opting out. And in order to make that happen, you need the funders to change their behavior, you need the journals, you might even need intellectual property in order to get the wagon moving. But I think it's possible, and I think it's in a small, highly visible field, there's a great chance to try doing something different. And given that I do think the future will be determined primarily by technology and the wisdom with which we develop and use it, I view that as one of the most exciting possible things that we can do with Gene Drive, is just to try a new way of doing science. Maybe it will fail. You know, it runs headlong against intellectual property incentives in many different fields, but there's not much commercial incentive to build something that is going to affect every member of a species, potentially. I mean, it's your one release and go. That's not a sustainable business model. So if there's not a lot of near-term commercial applications of this technology, that's then all the more reason to try the full transparency model here. In other areas, maybe, I don't know, synthetic virology, to just name a potentially dangerous one, you might not want every idea to be public, but you certainly want scientists to run their plans by other people. So I would love to see that be a sort of semi-open model in which you pre-register your experiments and run them by peers in different fields to see, is there an information hazard? Is there something we should be worried about? Is there a way that this development could be done and disclosed safely? So to me, the incentives are completely broken, but also you have to keep in mind, I'm at the MIT Media Lab. It's our job to ask very uncomfortable questions and try to change systems. And that's a blessing, but it also means I'm biased.
It'll be really interesting to see how that would be the most meta thing, I think, to come out of this whole Gene Drive discussion. I want to make sure we get to kind of one final question for Mel and Edward and Carrie before we go to audience questions. And this is about, you know, the challenges of regulating something that has a technology that's never been regulated before, namely where its goal is to spread genetic material. And, you know, I'm wondering how in the projects that you guys have been, you know, working on, how have you decided how much uncertainty communities are willing to accept, and especially if the risks and benefits aren't distributed equally, either, you know, geographically or socioeconomically? Like, how do you weigh everyone's input in a way that feels fair? Big question.
I think it's tough. I mean, for me, right now, it's just my goal. It's like my mission, just to get the community engaged. And if we can all be at a place where we know the risk, yeah, or know that there is some unknown risk, but that we're working with scientists who want to actually do field trials and try to measure the risk. But if we get the community involved, I think in our case, we're very lucky that, I mean, the risks seem very low right now. I think when I think about the Mice Against Ticks project, and the benefits are like through the roof. But we have to get the community engaged, and we're going to have to vote on how we think that the benefits outweigh the risks. And I just am grateful that we're working with scientists who are going to actually try to measure the risks and answer questions for our community so that we can make an educated vote on it.
Sure. I think the benefits outweigh the risks. Given that all the technologies that have been introduced to people, like the quinine, the coatam, and the rest of that, have worked, yes, but they have not eradicated malaria, for instance. And I think we would need some new technology now to go over this.
So I guess we have traditional ways for making decisions as well. We know we have our tribal structures and our formal ways of making decisions, but we also have, like most Indigenous people, large amounts of our people who have been disenfranchised from the tribal system. Colonisation has made them urban folk and disconnected them from the culture. And like most Indigenous people, we have a penchant for social activism. So when we don't like things, we're quite happy to tell people we don't like it and to protest and tell the whole world. So I guess having the discussions up front is really important, and having them in our places, having them, and I think that was echoed by the panel before, making sure that the conversations are in our environment and are held in our ways. And so for us, that's face-to-face and with lots of food. And so having those conversations is really important so that we can start to think about, I guess, how we would lobby for regulations that include our values and our ways of being and the safeties that we would want to see. By and large, Māori are quite cautious, and so I suspect that we have a saying in New Zealand that what's good for Māori is good for everyone. What's good for everyone is not good for Māori. So because we are quite cautious, usually if you can create a regulation or policy that appeals to Māori, it's usually, quite cautionary and appeals to everyone. But those conversations need to be had at multiple levels for us, and that's about making sure that you have all the right people there and you're talking to the right people.
Great. Well, I'll just turn it over to Julie. I think she's got some audience questions. All right.
Megan, I'll just flag, there's some that were long. I have the full version on my screen, so if you want to go to those, I will read those to you.
Okay.
The ones that go ...
Okay, well, let's just start at the top. So how can engagement with wealthy, educated, elite communities like those on Martha's Vineyard be a good model for working with more diverse communities? That's a good question about how generalizable your strategy is, and probably a good lead into talking about how every strategy should be different.
So I do think that my job might be a little easier than in other places because I am dealing with educating a community that has some framework of education to go with, and I think I think that my strategy is probably going to be different than the strategy of other places. But for us, we still have our own issues that we're dealing with. The summer community on Martha's Vineyard, for instance, might be very wealthy, educated, elite, but the actual community that lives on Martha's Vineyard year-round, we're mostly blue-collar workers, and we benefit from the visiting of, our population goes, for instance, right now at the beginning of our season, year-round population of 15,000 is now reaching, about 150,000. And so many of the people who are on the island in the summer are not year-round residents, so they would probably not get to vote. But that doesn't mean that we don't want to educate them about the project. And so we're planning on distributing informational pamphlets in the summertime so that even our elite and wealthy visitors can be informed of the project. But by and large, our year-round community is a very, very different demographic. But I think for us, it's just the key is, and I think we're going to really be successful in getting the students involved and getting the community involved by involving their children. We have one high school on Martha's Vineyard, one public high school. We also have a charter school, but one public high school where most of the kids funnel through. So it's a really good venue for educating and engaging the community, I think.
I feel like that's a good lead in to actually talk about the third one up there about how you define consensus permission. And I feel like this is especially interesting if you have communities who are people who are part of a tribe, but they are not co-located geographically. Or also, I think, how do you define consent permission, whether it's written or verbal, if you're in communities that have a cultural tradition of a verbal permission. So can we talk a little bit about how you go about defining that?
Great.
Consent for us is often verbal, so a vote wouldn't happen. 51% is definitely not consensus for us. We typically go for 100% consensus, and so we run what we call wānanga, where everyone comes together. The community would come from out of town. They would come to our marae, so our traditional house, and they would stay and they would argue it out until we had consensus. And they would go all night and all weekend and all week if they had to. So by and large, we aim for, 100% consensus or a decision that has the majority of the vote.
Yeah, I think for us, as I said, we did work with the communities of knowing what would be the right level of acceptance. So we decided not to decide ourselves, but to ask in each country, each community, what would be the right model. But it was quite interesting about the verbal versus written. We had this big discussion in Burkina Faso, which is a country where our research is most advanced, because traditionally they're using verbal consent because they believe that if you need it to be written, that means you don't really trust their words. But at the same time, we had the pressure from an international audience to be able to prove the consent. And so we had this really long discussion with this community trying to explain both perspectives, them telling us what is their perspective on verbal consent, us telling them what's our perspective on written consent. And so we explored many avenues, filming consent and so on. And at the end of this, I think, 8 months discussion, they said, okay, between us, we know it's a verbal consent, we trust you, but we also know there is this kind of imperative from the global public, from the global world of wanting to be reassured that we knew what we were signing for. You had told us what you were proposing, but also risk and benefit. And so we ended up agreeing on the form of written consent, but that was a consensus after a really long dialogue. So I think for me, it really represents this idea that you need to really just build, the models need to be built really specifically. I mean, we work in 3 countries and they have different models. So, I have one of my colleagues here, and I couldn't do it. I'm not doing it. It's all the colleagues in country who know very well the culture who are doing it, and that's how we manage.
Great. Well, I'm being informed our session is up, so I just want to thank our panel. I feel like we only just scratched the surface of some of these questions, but I'm really excited to see where all these projects go and how they shape the processes and set standards for the technology. So thank you. Thank our panel for joining me. Thank you.